Hi, everyone. I am Dr. Jeff Stratford from Wilkes University. Unfortunately, if you're listening to this, I am not feeling so hot. I get my shot in about an hour and 15 minutes, my second shot, and it looks like a lot of people are having some bad reactions and much better than being sick, but still, if you're listening to this, I don't feel so well. Hopefully I'll be here in person, but we'll see. I want to set the stage for this talk. I examine the effects of forest fragmentation and deforestation on tropical birds. And I just want to outline where the tropics are and just kind of set the scene. So there are tropics in Africa, equatorial Africa, Southeast Asia, and in Central and South America. So the neotropics where I work are in South America and Central America. The Amazon, where I primarily did my work, is located in the northern half of Brazil. So that's huge countries, Brazil. And this is the Amazon River running through the center of it. And you also have Amazon forest in the southern half of countries like Venezuela, Colombia, Peru, and Ecuador. This area here is actually a mixture of desert and scrub, and that separates this tiny patch of rainforest along the Atlantic here, and this is called the Atlantic rainforest, right? And this is Rio and Sao Paulo are here. You may have seen the cartoon movies, the animated movies, Rio. So the biggest threat to wildlife, including birds in the Amazon, is deforestation and forest fragmentation. Deforestation is the flat out removal of forests. So once there was forest and now there's something else. Forest fragmentation is in the process of deforesting, patches of forest are left. Forest fragmentation is a little bit more insidious than deforestation because as you might expect, if you remove habitat, the animals can't live there. Yet forest fragmentation leaves patches of what looks like habitat, but yet maybe missing many of the species that live in forests. Forest fragmentation in the Amazon, and that includes essentially all over the world, the same process that creates forest fragments, is due to agriculture and urbanization. So these are two sites in the Amazon between the port city of Manaus and Venezuela is a bunch of cattle farms, and just whatever process or the idiosyncrasies of the landscape will leave these patches of forest here, and it'll be surrounded by agriculture. And the agriculture in the Amazon is primarily cattle farming. So you'll have these patches here. When it comes to urbanization, what happened is you have this, if you go back a number of years, this small port town of Manaus, which supported the rubber trade. And it grew and grew and grew, but as it grows, for whatever reason, you are left these patches of forest here and here and here and so on. So this is a pretty common pattern we see throughout the world in the formation of forest fragments. We know about forest fragmentation from two projects primarily. The Biological Dynamics of Forest Frogs project, that is located in the central Amazon, and it was established in 1979. It's still active by the World Wildlife Fund and coordinated by Tom Lovejoy, who's Professor Emeritus at George Mason. Currently, the Biological Dynamics of Forest Fragments project is managed by the Smithsonian and INPA is the Brazilian version of the Fish and Wildlife Service, essentially. This is a project that started when Brazil had incentives to both preserve forest and deforest. So there were incentives to deforest in the sense that they wanted cattle farms, particularly north of Manaus, to set up an area where there were, to make the Amazon a bit more productive. It was also a time when they had incentives to not deforest, because if you were given so much acreage, 50% had to remain as rainforest at the time. And so what Tom Lovejoy did is he said, well, if you're going to leave 50%, we can actually set up an experiment. So with coordinating with farmers, what they did was, as they deforested, they left these wonderful experimental plots of 1,000 hectares, 100, 10, and 1 hectare forest plots. And these are replicated. So this just shows two, this is a 1 hectare forest plot, this is a 10 hectare forest plot. And there were other ranches where the same setup occurred. And one of them, actually two of them, have 100 hectare forest plots. So we actually have this huge landscape scale experiment with controls and with replicates. And from that, this project has created hundreds of papers, if not thousands by now. The other project is the Smithsonian's Barrow, Colorado Island Experimental Station. And so that was formed when the Panama Canal was formed. And so this strip, what you're seeing here, was actually all forested at one time. And when they built the Panama Canal, they had the lock system on the Pacific coast and the Atlantic coast and dammed it up. And that produced a lake, and that is Lake Gatun. But the mountaintops in that lake became islands. So Barrow, Colorado Island is essentially a mountaintop. And this has been studied for decades and decades and also produced hundreds of publications. So as a result of several studies, it looks like the epigeial insectivores are particularly sensitive to deforestation and forest fragmentation. They used to be called, by the way, terrestrial insectivores, and I might be the only person in the world that calls them epigeial insectivores. The reason I call them epigeial insectivores, I think it's a little bit more descriptive than terrestrial, because I think most people, when they hear terrestrial, think the alternative is aquatic, so aquatic and terrestrial. But terrestrial here means they forage on the ground, as opposed to arboreal, which is in the trees. Epigeial means that they are foraging on the surface of the soil. So these birds here are all insectivores. And so what they do is they walk around and pluck insects off the leaf litter. This bird on the far right is wing-banded antbird. And this bird right here actually will flip leaves up in the air. So you can actually watch this bird from a distance. It'll throw leaves up in the air, then quick dash and try and catch the insects that were underneath the leaves. So a number of years ago, Phil Stouffer and Rob Beauregard started monitoring birds from year 0. So as the forest fragments were created, and then follow them through time. So what they found was terrestrial insectivores are lost from 1 and 10 hectare forest fragments. So they basically, you find about 10 to 20 species, right, in any particular plot. But as you go through time, those forest fragments, once they become isolated, lose species and they don't really come back, right? So this is 9 years afterwards. I know that these are still being monitored, and many of them still to this day do not have most of those epigeial insectivores. I went in a few years after this study was published and said, well, an alternative explanation is this might be due to regional declines. So did we really show an effect of fragmentation? So what I did was I set up controls. And so what I had available to me was a very large continuous plot that had a whole system of trails set apart by 100 meters. So we had a 100-meter up by 100-meter grid set up in continuous forests. And I used that grid to create 1 hectare plot. So one of those squares in that plot was 1 hectare. Right? So I mapped out a 1 hectare plots, 10 hectare plots, and 100 hectare plots. And I compared that to actual forest isolates. So you can see what happens is as you sample more land, you sample more birds. And that's true if you are fragmented or not. So both these lines are increasing. So the more you sample, the more you get. That is not necessarily surprising. But here I show there is a very distinct signal of fragmentation. So when you sample 1 hectare control plots and other control plots, you just have more species than equivalent sized forest fragments. So this shows the effect of being isolated. So I finished my master's and go on and do a PhD with Doug Robinson. And I decided I really want to figure out what's going on with these birds. Why are they so sensitive to forest fragmentation? And we wrote a paper and outlined a bunch of hypotheses. And we had to create a new set of hypotheses. Although people have been working on forest fragmentation for decades in North America, we thought that the mechanisms of bird loss in the tropics were different. So the epigeal insectivores in the tropics are primarily antbirds, and there are no antbirds in North America. So if you talk to a North American ornithologist, they would point to nest parasitism and nest predation as the primary mechanisms of why birds are missing from temperate forest fragments. But in the tropics, we don't have the same brood parasites. So there are cowbirds, but not like brown-headed cowbirds here. They tend to be specialized in the tropics. And the nest predators here, which include things like foxes, raccoons, possums, those are all species that are either in lower abundance in tropical forest fragments or just absent. So these things don't work when you go to the tropics. And we wrote this paper that made an analogy to Gulliver. And if you ever read Swift's book, what happens is this English surgeon gets on a boat and he has terrible luck and he keeps ending up on these different islands where life is completely different, and his ideas of how life should work, because it works in England, does not work on these islands where he's either very tiny or very large. And we thought that was a good analogy for what was happening in the tropics. So we came up with a bunch of hypotheses, and the rest of the talk I will sort of say, like, following up with that, what kind of support do we have for each of those hypotheses? So we proposed that maybe terrestrial insectivores need large forest fragments, right? Maybe the small forest fragments are too small. Phil Stouffer, who was my master's advisor, he radio tagged a whole bunch of birds and then looked at how much space they needed. And it ends up that most of these birds need 4 to 7 hectares of forest. So that explains why they are lost from 1 hectare forest fragments, but doesn't explain why they're missing from 10 and 100 hectare forest fragments. So they have the space, they're just not there for whatever reason. So this partially explains that pattern, but not fully explains. All right, then the other idea is more physiological as well as behavioral, is that maybe forest fragments are too hot. So forest fragments, one of the reasons why they're different than just large patches of forest is edge effects. And what you have is the sun penetrates forest edges, especially hard edges such as those when forest meets pasture. So if you can imagine over the course of the day, the sun is going to be able to reach the bottom of this forest fragment, this 1 hectare forest fragment, from several sides as it goes overhead, right? And in continuous forest, because the canopy is so dense, it's well shaded. And we've seen where I actually took temperatures, and we were getting 38 degrees all the way up to 42 degrees in exposed areas. It's very uncomfortable. It's well over 100 degrees. However, in shaded areas, it's 27 to 30 degrees constantly throughout the day, which is really nice. It's very pleasant. And so if terrestrial insectivores either behaviorally avoid these places, or maybe they can't even like metabolically handle these sites, that would explain why they're missing from smaller forest fragments. What Henry Pollock did as a graduate student is he mapped out microclimates. And so he ran around in continuous forests and took temperatures of where these birds were using and mapped out, what these birds use in continuous forests. And what he found was that the Stratford-Robinson microclimate hypothesis was not supported. So it goes. We got shot down there. But fortunately, you come up with more hypotheses. So we gave them more targets to shoot at. The microclimate hypothesis that was tested by Pollock looked at how birds use continuous forests, and it was like a behavioral response. What we don't know, and what's still out there to be tested, is sort of a physiological response to elevated temperatures. It's one thing if a bird passes through a hot patch. It's another thing if a bird has to persist and live in a hot patch. And there's been very, very few birds put in metabolic chambers to look at upper critical temperatures. And most of those birds are the oddballs. So these are the birds that live in fields and live in these 40-degree Celsius environments. And those are remarkable birds. But forest birds that like cooler environments have been completely ignored. So the other hypothesis that we looked at is, are they too bright? One of the things about these birds, these epigeal insectivores, is that they're living in a dark environment. They are searching for brown insects on a brown background. And so they must have just amazing visual acuity to find like a cockroach on top of brown leaves in a relatively dark background. So one of the hypotheses is just like that they're too hot, maybe they're too bright, because light can penetrate in from all the sides. So maybe these environments are just too bright for these birds. And you can even see, like in these two species, they have relatively large eyes. And so this was tested by Cameron, who was a graduate student of Phil Stouffer's. And they basically captured bird and then looked at the size of the eye, the diameter, relative to the whole body size. So a small bird with big eyes would be more adapted, and you would predict to be more sensitive to fragmentation. However, what they found was no support for the it's too bright hypothesis, right? But what they said is, just from these eye measurements, it doesn't look like this hypothesis works, but what we need are behavioral experiments, because maybe they're just avoiding bright areas. And that was done. So Mike Patton did what Henry Pollock did, but not measuring temperatures. He looked at light and looked at the light environment used by neotropical birds. And he actually did find that birds avoided bright environments, right? So it's may not necessarily be a physiological consequence of eyes, but it may be like a behavioral response to bright environments that they're just avoided. And this would explain why they're not necessarily using smaller, brighter forest fragments. Okay, I looked at the habitat hypothesis for my master's thesis, and what the idea is if these forest fragments are brighter, then vegetation has more light and you should have more essentially lush vegetation. So the question is it too thick for epigeal insectivores? So I ran around these forest fragments and measured vegetation, 28 variables, and had hundreds of samples. And then I also compared these to sites where I observed birds actually foraging. So we have vegetation of where birds forage compared to what's available in forest fragments. And then I also had random sites and continuous forests. So here are my results. And what we found was as you decreased forest size, that plant cover, so this is 3 to 10 meters. So this is, if you drop an imaginary line from 3 to 10 meters, if you'll hit vegetation, right? The far left is continuous forest, and you can see that most birds on the right here fall within that range. So they're very comfortable with sort of more open vegetation at only 3 meters. So this would be relatively open habitat. But as you increase or decrease forest area, they get more and more dense in their interiors with vegetation. And you can see that in 10 hectares and 1 hectares that these places have vegetation that's too dense. And we also looked at, leaf litter depth, and what we found was the same thing, that as you decrease the area, the leaves get thicker and thicker. And that's because you have lots of leaf turnover. So you have rapid growth, and plants tend to drop their leaves after they grow. So you only find leaves at the ends, right? And so as you get rapid growth, you get rapid turnover of leaves. So you tend to accumulate leaves. And most of these sites actually have leaves that are too thick for these epigeal insectivores. And in fact, if you compare the reference site, so continuous forest leaf litter, those sites that birds use tend to actually have thinner. So they are selecting very thin leaf litter in continuous forests, and forest fragments have very different. So I'll conclude and say that we have pretty good support for the vegetation hypothesis. So but that support is indirect. And what I mean by that is I'm just looking at where birds forage, what would be really great, and we're guessing what variables are important. What we really need are experimental manipulations of vegetation. So somebody needs to go and remove vegetation or plant vegetation and see how the birds respond. And that would be a great experiment that could be done. And it would also, affect the light environment, so you could knock out two hypotheses at once. All right, the newer stuff is this dispersal limitation hypothesis. And what that means is, and what we've seen is that, birds, it looks like, may not be able to colonize these forest fragments, but we're not sure, right? There's something about them that they're not using. And we come up with two parts to this, that physiologically they're unable to reach. So if you imagine this is a hot pasture, I am standing at camp, but there's forest right behind me. And if a bird wanted to colonize that forest fragment, it would have to fly across this hot pasture. Maybe they just don't have the flight muscle to make that trip in one bout. So if they stop, they essentially get too hot and don't make it. Or perhaps if I'm standing in a forest and looking across this pasture, they say, we are not going across that pasture to get to that forest. So that forest might be fine. We are not crossing, and we don't like cows, we don't like open areas or whatever, that they're just psychologically unwilling to cross these open habitats. So as I mentioned before, temperate birds and tropical birds are very different. North American birds, here's a study that looked at a patch of woods over a number of years, decades actually, and compared that to Barrow, Colorado Island, where Doug Robinson took over for Frank Chapman of the American Museum of Natural History. And so they have continuous surveys from the 1930s, and Doug's actually still doing these surveys. What you find is this remarkable difference between these two places. In the temperate zone up on top, the species identities are not that important. What's more important to see is that there's gaps and then they come back. And so, for example, the bottom two, white-breasted nuthatch and Carolina wrens, they're there, they're not, they're there, they're not, and so on over the years, which implies that this woods in Illinois, that the birds disappear for whatever reason, and then a bird flies there, recolonizes, stays for a year or two, then it disappears, gets recolonized by different individuals, right? That's important. Different individuals are colonizing and then disappearing. And that gets repeated over time. So you end up with these dashes. If you compare that to Barrow, Colorado Island in the Panama Canal, what you get is birds are there and eventually they peter out and disappear and they don't come back, right? So there was a number of birds that were there when Frank Chapman was there and are no longer there, despite decades of them being able to colonize. So this was what I consider a remarkable result. And so people have looked at the psychological unwillingness to cross open areas. And Susan Lawrence, she did a really clever experiment, and she grabbed birds and took them across the pasture and let them go and see what they would do. Would they fly back across? And it ends up that in a control forest, if you move them away, they can actually navigate back to their release point pretty quickly, actually. But if you take them across a pasture, the fragmentation species, they won't cross large gaps for whatever reason. They just won't. They'll cross, some of them will cross smaller gaps. So she took them across one highway and they were able to cross about 75 meters. But these pastures, the forest fragments that are separated by 250 meters, that actually creates a situation where birds see that as something they just will not cross, okay? Then later on, Randy Moore, working with Doug Robinson, did some more release experiments, except they were very interested in what is the actual ability of birds to fly. And so what they did was, working on Barrow, Colorado Island, they captured birds on what I'll call the mainland, not on BCI, but on the mainland, took them in a canoe, released them, and see how far they could fly, right? Because these are not swimming birds, right? So they would fly, and if they poop out, they just fall in the water. So he did this release experiments. There was somebody in a motorboat with a net, scooped them up, and would bring them back to the mainland. But this gave you the physiological limits of their flight ability. And what's remarkable is that some of these birds, so these are all ant birds, and includes fragmentation sensitive and fragmentation insensitive species. And what you can see is some birds cannot fly 300 meters, right? Compare that to temperate birds, many of which migrate and will fly several states on their journey to Central and South America. These are South American, Central American birds that can't fly 300 meters, right? Other birds, no problem. That Faithornis is a hummingbird. Pipra is a small manakin. It's a fruit-eating bird. Same with Mionectes. So these birds are moving around the forest, searching out flowers and fruit. The insectivores, however, just incredibly weak flyers. So what we notice is that there's a group of good flyers that can fly greater distances, right? At least 1,000 meters, and then a group of really poor flyers that sometimes they can't even fly 300. Some can't even fly more than 100, right? That's how physiologically constrained they are in their flight abilities. And these are what we call single bout, so a single flight from one point. I have a collaborator, Brian Rourke from California State Long Beach, and I was able to get him some muscle from some museum collections. And so what they did was, him and a graduate student, Kelsey Condell, what they did was looked at myoglobin concentration in the heart and found that, and in pectoralis, and in the heart, there's no difference between good flyers and bad flyers. So similar concentrations. However, in the pectoralis muscle, which is the major flight muscle, that good flyers had much higher concentrations of myoglobin than the poor flyers, which had overall much lower concentrations of myoglobin. So again, these are birds that primarily walk around the forest looking for insects, and really the only time they would fly is from a predator. So one cool thing is we did a regression of myoglobin concentration versus the distance they could fly. And what you find is a very strong correlation between single flight distance and the concentration of myoglobin. So this is one of the more remarkable results, and they are now working up the manuscript right now. So let's summarize. Why are these epigeals insectivores so sensitive to forest fragmentation? They do require at least 100 hectares of forest patches. They are unwilling to cross gaps. Okay, they are unable to cross gaps, especially over water, right? So Barrow, Colorado Island likely never be colonized by some of these species. They don't like the habitat in patches. So even if you transplant these birds there for conservation measures, they'll find habitat that's not appropriate for them. They don't like the hotter microclimates, so they can move through it, but if they're stuck in there, they won't use it. It looks like they're unable, so that hypothesis that they're unable to use brighter microclimates, it doesn't look like that hypothesis works, but they are quite happy to use the so they can use it, but they just don't like it. So that's the last hypothesis. So it looks like in our Gulliver paper written in 2005, we did okay. We sort of have some mixed results, but overall, it's kind of not one hypothesis, but it ends up like a whole suite of hypotheses actually fit why these birds do not persist in smaller forest fragments. So what does this mean for conservation? Well, for one, you have to have forest reserves that are much larger than 100 hectares, okay? Because you need to overcome the edge effects. So small forest fragments, 10 hectares and smaller, will be overwhelmed. The vegetation won't be right, be too bright, too hot, and these epigeal insectivores will not use them. They also need to be connected to other forest reserves. So you can have a large forest plot, but if it's not connected by some sort of forested corridor, birds aren't going to use it because one, they are either unwilling to cross open areas, or two, they are just simply unable to reach these forest patches. So it means in conservation of the species, you're really going to have to plan out appropriate measures. I would like to thank the people that did the physiology work. So Brian and his graduate student, Kelsey, did the myoglobin extraction, and well, Brian at least is continuing to work on more samples. Randy Moore did the release experiments in Panama, and Doug Robinson is the PI and kind of roped all these people together. So with that, I'll stop. I think I'm a little bit early. My Alexa has not reminded me that it's been time. Unfortunately, I'm not here. So, but feel free to reach out and ask questions. This is the passion, right? Is neotropical birds. I work a lot here in Pennsylvania, but if I get to the tropics, that would be great. And I'd love to collaborate with anyone that sees any more ideas. So these are just the hypotheses we came up with. There's probably a few more, and it'd be great to explore them. And with that, let me stop sharing. And let me thank you for listening. I appreciate all of your time, and I hope you enjoy the rest of the symposium. And with that, I'll say goodbye.